Good morning again. This will probably be the last set of videos. I'm going to go over the renal system, kind of combine structure and function in one thing, kind of one set of videos. So that'll be lab, lecture stuff combined. The exam, it'll be some multiple choice and it'll have a few identified for the structure. So I'm kind of combining the last exam to cover a little structure, a little function on the renal system, because that's honestly all we have time for. Digestive system, go ahead and read through that chapter as well. You've got the Learn Smart. That's all I'm going to do with it because I have to be wrapped up by Friday. So you can figure the exam will be posted Thursday maybe. So we don't have too much time. Try and get through it. Then I'll calculate your final grade. Again, the way I'll calculate your final grade. On Blackboard, you have a percentage for the in-class stuff. It's already finished. It should be posted up there. You got a percentage. That's going to be 60% of your grade. Everything I do online, all the tests, all the Learn Smart, all the quizzes, I'll take an average for that. The way you do that, take your number of points that you get, divided by the number of points possible, that's your percentage. Then I'll just take the percentage in class, multiplied by 0.6, the percentage outside of class, multiplied by 0.4, add them together, that'll be your final grade. So that'll be done, have to be, I think, submitted next week. So what I'm going to do today, first, I'm going to go through kind of the general functions of the renal system, a little bit of its basic anatomy, then we'll go through its function a bit. I'm going to start off, I'll do a little video on the small screen just to list the functions, then I'll do some scribbling, and I'll go back to some diagrams on the small screen, and we'll look at those. Take care for a minute. Alrighty, these will be the functions, kind of the major functions. I grouped a few of them together for the renal system. You got one up here. This is removing nitrogen waste. Nitrogen waste primarily comes from metabolism of proteins. You remember the protein has that amino group and then a couple of carbons left. The amino group cannot be used. So if you burn a protein, you can use a 2-carbon bit, basically run it through the Krebs cycle, use it to make ATP. The amino group is toxic, has to be removed. It initially comes off as ammonia. The liver will turn into urea, which is a little less toxic, but it is still toxic. That's, what we generally call a nitrogen waste. That has to be removed. Then, a bunch of other functions. We've talked about a few of these a little bit with the endocrine system, but control blood concentration. That is the water content in the blood. That is controlled via ADH. The kidney can eliminate excess water or retain water to keep your blood's concentration at 300 milliosmoles. Number 3 here, control blood volume and blood pressure. That's aldosterone. We talked about that a little bit with the heart and a little bit in the endocrine system. Aldosterone, when it's released, it targets the kidney and it makes the kidney reabsorb sodium and water, which primarily boosts the blood volume, which will boost blood pressure. Blood volume and blood pressure are related. Blood volume goes up, blood pressure goes up, and vice versa. Number 4. I think I mentioned this with the circulatory system a bit. The kidneys also monitor blood oxygen levels. And if blood gets a little too low in oxygen or hypoxic, it will release renal erythropoietin, and that's what increases the synthesis of red blood cells. So it will increase your hematocrit. Other things, controlling blood pH, hydrogen and bicarbonate ions, that's an acid, that acts as a base. The kidney can selectively release either hydrogen ions or bicarbonate ions to keep your blood pH at about 7.4. Then controlling electrolytes or ion levels, particularly sodium and potassium, which are really important for those excitable cells like neurons and muscles and so forth. So the kidney can excrete sodium, excess sodium, or retain or secrete potassium as needed. And then I just have a random catch-all category here, other. That's basically breaking down proteins, deamination, detoxification of certain chemicals, and a few others. So that's the basic functions of the renal system. Then, go to my next slide here. Hit this one real quick. The anatomy. I don't have a good diagram or model. I'll have to get a photograph out of the lab. This will do. This is showing you the main components of the renal system. I'll go over them on the board in a minute. Not that much to it. You got the kidneys, which I should mention, the kidneys get a huge amount of blood flow. They're like, 0.4% of your body mass, but they receive, somewhere between 20 and 30% of your cardiac output. So a huge amount of blood goes through these renal arteries to the left and right kidney. So a lot of blood supply for their size. And of course, that's because as the blood passes through the kidneys, they're going to process it, remove unwanted material like excess water, excess salt, or urea. Now, as the kidneys remove excess material from the blood, you got to do something with it. So the excess fluid, the waste, will go down these little tubes. There's one from each kidney. These are small muscular tubes called the ureters. They will drain this waste fluid from the kidneys into this little smooth muscle bag right here. That is your urinary bladder. You store it in there sort of temporarily so you're not dribbling a trail of urea behind you. It would make it easier for predators to find you, you know. But that's a storage side. I think it can hold up to about half a liter. And when it gets full, there's a third opening here. This is the urethra, which goes from the bladder to the external environment. And you can contract the smooth muscle in the bladder and squeeze all the waste out. So that's kind of the gross anatomy. That's really all you need. Kidneys, ureters, bladder, urethra. What we'll emphasize for the structure, we're going to separate this kidney out, kind of cut it in half and look at the internal parts. And we'll actually have to look at the microscopic structure to figure out how the kidney does what it's supposed to do. So that'll do it for this bit. All right, let me get started again. So we went over the basic sort of functions of the renal system, primarily the kidneys. The rest of it's just plumbing, ureters, bladders, urethras, just a way to get it out of the system. So let's look a little bit more at the kind of the fine detail characteristics of the kidney itself. We'll just look at one. So imagine, here's my one kidney. I'll kind of make it big, and I'll shrink everything else a bit. There's your kidney. And a couple things about the kidney. They are embedded, you have two of them, kind of right and left. They're embedded kind of in muscles at the dorsal side of the abdominal cavity. They are behind the parietal peritoneum. Again, most of the abdominal organs are covered by the visceral peritoneum. Kidneys are not. They actually develop behind the peritoneal membranes. They refer to that as being retroperitoneal. So they have some of their own protective coverings. There's some connective tissue kind of holds them in place. There's often big fatty pads around the kidneys to protect them because they actually project a little bit below the ribs, especially on the right side. The liver kind of pushes the right kidney down a bit. They're kind of sensitive to damage back there. So there's a pad of fat around them to give them a little protection if someone, I don't know, kicks you in the back. And then there's also its own sort of connective tissue capsule around the kidney. Again, a little bit protective. So no peritoneal membranes on this one, just a fibrous capsule on the outside. And if we look at the kidney's sort of gross anatomy, I'll look at some prettier pictures here in a bit. But when you section a kidney, there's an outer layer, might be about a centimeter thick, on a pig kidney, human kidney, a little smaller. This outer layer is the cortex. Again, a lot of organs, it's the outer layer, just means cortex. Had that in the adrenal gland, had it in the brain. Cortex has kind of a granular appearance. And then there's a middle portion of the organ. Of course, this is called the medulla. And there's a few interesting characteristics of the medulla I should mention. The medullary tissue is arranged as sort of little triangular blocks. So the medulla is made out of a bunch of things called medullary pyramids, because they look like a bunch of little pyramids in there. So it's a little issue in the lab, you know, are you talking about the layer here, or you're talking about the pyramid? We don't need to worry about it because we're not going to have that kind of lab exam, but the medulla is the inner layer, but it's made up of these triangular blocks called renal pyramids. Now the cortex does dip down between the pyramids. These are what they call in lab the renal columns, so there's some cortical tissue that kind of projects down between the pyramids. And then, of course, what the kidneys are going to do, they're going to remove waste material from the blood, kind of a place to put it. So there's a series of sort of open cavities or spaces in the kidney. These cavities or spaces are generally called calyces. And what you'll find is right around each pyramid, there's a little open space. So right in front of this pyramid, kind of the little tip of the pyramid here, this bit here, and here, and here. These are specifically called renal papillae. A papilla is kind of like a little finger-like projection. So there's little bumps projecting out into these hollow spaces. These little hollow spaces, That's what they refer to as a minor calyx. Minor calyx is a little tiny pocket or open space in front of the papillae. As we'll see, as the kidney processes the blood and removes waste material, that waste material will end up dribbling out some little holes, little ducts at the end of these papillae, and it will fill up these minor calyces. And then, kind of like a river system, the minor calyces will flow into bigger spaces. Be several of them, but that is a major calyx. I'll look at that on a small screen in a bit, a little prettier picture than my nastiness up on the board. But that is a major calyx. So the waste material, what we're going to start calling urine at this point, dribbles out of these papillae into the minor calyces. They flow together in this major calyx, and then there's a little funnel-shaped bit. That's usually what they call the pelvis, renal pelvis. It's like a funnel. It will drain the urine out into the ureter I already mentioned. So that's the ureter coming out there. Those are kind of the bits we need. Now, when you look at the kidney, again, I give you a big list of what it does. It's a tremendous amount of different functions. To understand how the kidney does most of what it's supposed to do, we have to look at the microscopic structure. This is kind of the gross anatomy. If I just took a kidney and sectioned it, opened it up, you'd see these parts. That's what we usually identify in lab. The cortex, the pyramids, the columns, the minor, major calyces, pelvis. You can see all that with your eye, but that doesn't help us explain exactly what's going on. Turns out if you look at high magnification at the kidney, you'll find it's made up of a series of microscopic tube-like structures. That's what we want to look at. These microscopic tube-like structures are called nephrons. I think if I remember, there's like 10 million of them in each kidney. So you got a lot of them, they're microscopic. It's kind of like looking at the sarcomere of the muscles, in Anatomy 1, when you want to understand how a muscle generates tension, you look at the level of the sarcomere. It's kind of the functional unit of a muscle. For the kidney, the functional unit is the nephron. If you can understand how the nephron works, the kidneys are no more than millions of those kind of working collectively. So we're going to kind of zoom in on the function of the nephron and kind of what it does to allow the kidney to do all of these things. Now, where the nephrons are located, I'll have to draw a separate nephron in a bit, but basically the nephrons, they typically start up in the cortex. Again, it's a series of tubes. They run through the cortex. Then there are portions of them that loop down into the medulla in many cases, then back up into the cortex, and then back through the medulla. So there's going to be about 1 million of these small microscopic nephrons. Now what we're going to do, I'm going to basically pull out one nephron for the next part of the video. We'll look at the pieces of the nephron and kind of what they do in general. And we'll also have to look at the blood supply. All of this process is taking blood and removing junk you don't want. So we need the blood supply here. So we'll look at the nephron and we'll look at the associated circulatory system of the nephron to kind of figure out how the kidney does what it does, and we'll kind of just split it up into some basic parts of kidney function or urine formation. Let me go back to the small screen now and just kind of show you a model of the kidney that's going to give you a better picture than the mess I put on the board. All right, here's a better picture of the kidney. This is a cross-section through a kidney. This is a ureter coming out here. Now what I want you to note is this bit up here, kind of right along here, right along here. This is the cortex. I'm not even going to try to write it on there with a mouse because, I can't write with a pencil hardly. But that's the cortex there, that layer right there. They're kind of showing it with a different texture. These big blocks right here, kind of triangular looking things, that is one renal pyramid of the medulla. That little bit there, that is the papillae. So you got the renal pyramid and you got these little papillae sticking out. So there's a pyramid here. That's its papillae right there. Lost my picture. That's a papillae there. You got a pyramid here. That's its papillae. One here, one here, one here, and so on. So these are all pyramids of the medulla. And you can see the cortex dips down right here. This area, that's one of those renal columns where you get cortical tissue dipping down. Then here are the calyces. This little hollow space right here, I'll use that one, it's best. That's a minor calyx. There'd be one here, be one here, one here. The minor calyxes will collect the urine. It comes together and it goes to this larger space here. That's the major calyx. And there's your little pelvis coming out, draining into the ureter. Now the nephrons, at this magnification, you can't really see them. They're showing this bunch of little tube-like things here as part of the nephron. We'll look at that in more detail later. And while I'm here, let me look at the circulatory system so we have it on a good picture. But this is that renal artery. We've already identified that in the lab. It comes into the kidney. There's your renal vein. Once it comes in, it splits. So let me run through this quickly. This is a segmental artery. So the renal artery splits into segmental arteries here. That one will then split, and you have these arteries running up between the pyramids. Arteries and the veins basically have the same name. So that's renal, segmental, which will then split into, these are interlobar arteries. They kind of run through the renal columns here, interlobar, interlobar. Then they'll arc around the back, where they arc around the back of the pyramid, like right here, all of these back here, kind of running between the cortex and the medulla, all these right here. So like right here is a good example. Those are arcuate arteries. It'll be on your renal lab sheet. So the blood goes from renal, segmental, interlobar, arcuate, arc around the back. And then coming off of the arcuate, you see these little arteries running up here. Oh, that's a terrible line. Running up there. These are interlobular arteries, or they're sometimes called cortical radiate arteries. Two different names, same artery. Small arteries, those just kind of branch and go through the cortex. And now we can't see much else on this because we're getting smaller. But if we look right here, kind of branching off here, those are arterioles. Now, we've talked about arterioles with the circulatory system, but in the kidneys, the arterioles are given some names. And if I zoom into another picture, so I'm going to look at this little bit right here. I'm going to zoom in on the next slide and kind of that bit there, right? Coming off of that interlobular artery. Here it is. This would not be visible to the naked eye, but this is that arteriole coming off of the interlobular or cortical radiate artery. This is an arteriole, but it has a name. This is called the afferent arteriole. I'll draw these again on the board in a bit. And of course, this arteriole will then lead into a capillary bed. All of this red mess in here, this little ball-like knot of capillaries, that is representing a capillary bed. It's named, it's called the glomerulus. Again, I'll write it on the board in a bit. But this is a capillary bed. It's like any other capillary bed, more or less. Except here's where it gets a little bit different. Normally, draining a capillary bed would be a venule. That's not the case here. This thing coming out, this is another arteriole. It's an efferent arteriole. So notice this is a weird kind of setup for the plumbing. Normally, you'd have an arteriole going into a capillary bed and a venule draining it. You don't have that here. You have an arteriole going into the capillary bed, and you have an arteriole draining it. And I'll explain back on the board in a bit why that is. It has to do with maintaining very high blood pressure in this glomerulus, which is necessary for urine formation. All right, I'll stop there with that picture for now and get back to the big board. Okay, now go over the nephron a little bit and its blood supply. So here's kind of where we left off on that last little blip of a video. We have, give myself some room. Imagine this is the interlobular artery, the interlobular artery or the cortical radiate artery. That's the one that's coming up, through the cortex. Branching off of that, there's a small arteriole. I'm going to call it the afferent arteriole. Again, normally arterioles, we didn't name them, but in the kidneys, they're named because they have kind of a special function. Now, if we follow this afferent arteriole out, it will then split into a capillary bed. I'm just going to make a bunch of squiggles here to represent the capillary bed. And this is called the glomerulus. Glomerulus is the name of this little clump of capillaries. Now again, what's odd, you have an afferent arteriole going in, Coming out or draining this glomerulus is the efferent arteriole. Again, afferent means going to, efferent means going away, so the names are descriptive. That's a blood supply. Now, it's a little weird. Why an arteriole going in and out? That's an unusual setup. It's partly because if you look at every other capillary bed in your body, you have the arteriole, then a capillary bed, then a venule. The blood pressure in most capillary beds is quite low, 10 millimeters of mercury or so. That's because most of the resistance, remember arterioles, we consider those the major resistance vessels in the circulatory system. They have a lot of resistance, so after you go through the arteriole, the pressure has dropped tremendously. So pressure is about 10 millimeters of mercury in most capillary beds. But if you look at the setup in the glomerulus, the afferent arteriole is actually relatively wide. The efferent arteriole is the small one, smaller diameter, more resistance. So your resistance, I'll just put an R here, is higher in the efferent arteriole than the afferent, which means the reason that's there is the blood pressure in the glomerulus is about 60 millimeters of mercury. That's about 6 times higher than any other capillary bed. Now, the reason for that, remember, we're going to have to take stuff from the blood, the junk we want to get rid of, and get it out of the blood. How are you going to do that? This is a process called pressure filtration. Most of your capillary beds actually leak. You know, imagine if you run like water through a garden hose and someone's put a bunch of little pinholes in it, like those soaker hoses you might use in the garden. You run a high pressure fluid through a hose with holes in it, water squirts out. Well, capillaries are the same way. You're running high pressure fluid through them, it's blood, but they all have a little bit of leakage. You know, there's junctions between the cells, little pores, that sort of thing. That causes some liquid to leak out. Not cells, not big proteins, but a little bit of liquid. Now, all of your capillary beds, not counting the kidneys, they might lose 7-8 liters of fluid a day. That gets returned to the circulation through the lymphatic system, which I haven't talked much about. But the lymphatic system takes that fluid that's leaked out of the blood vessels. Lymphatic vessels then pick it up, and it's basically interstitial fluid. That's how it circulates. And those lymphatic vessels will bring it back to the circulation. In the glomerulus, because the pressure is like 6 times higher, it's like really cranking up the water pressure in a soaker hose. Fluid squirts out. So these glomerular capillaries, because of the high blood pressure and the fact that these are called fenestrated capillaries, they literally have holes in them. So they are like a soaker hose or something. When you run blood through this glomerular capillary bed with 60 millimeters of mercury pressure, fluid just squirting out like crazy. Now, where does that fluid go? Here's where we start, the nephron. Right around this little clump of glomerular capillaries, this is where we'll find the beginning of the nephron. I'll exaggerate it a little bit. This is called the Bowman's capsule. Now, it's almost like imagine you had a balloon and you punch your fist into it. That's kind of the Bowman's capsule. There's an outer layer, often called the parietal layer here. Then it folds back and it intimately covers the glomerular capillaries. It's almost fused to them. This is called the visceral layer of the Bowman's capsule. The visceral layer of the Bowman's capsule is made with a layer of, they're basically modified squamous cells. They're fairly thin, and the cells are called podocyte cells. They have these little finger-like or projections sticking away from them. They basically look like a screen. So what you have here, this layer, this is what you could call the filtration membrane. It's the wall of the glomerular capillaries and the visceral layer of the Bowman's capsule. The capillaries are fenestrated, have holes in them. The visceral layer of the Bowman's capsule, it's like a bunch of slits. I mean, if you looked at it high magnification, you'd see all these little slit-like openings there. So it's got a very, very high permeability. So when you run this high pressure fluid, blood, through the glomerular capillaries, it's high pressure, they're so leaky, fluid is going to squirt out. From the blood, it ends up moving into the lumen of the Bowman's capsule. It's like a little bag, it's a little collection area. This material that is moving out, it's just by pressure filtration. It's not terribly selected. Everything that moves out of the blood, in fact, everything moves out of the blood except the really big stuff. Those slit pores and fenestrations don't allow cells to move across, so you should never get any cells in here. No red blood cells, white blood cells, platelets for the most part. They're all cell components that are too big. Most of the plasma proteins are too big to move through this filtration membrane. So anything of large size won't get across. What does move across? Almost all of the ions like sodium, potassium, and so forth, all of the water, most of your nutrients like glucose, amino acids, and so forth, that moves into the filtrate. The filtrate is basically the same composition of blood, minus most of the proteins and the cell components. That's where we're going to start urine formation. So the Bowman's capsule is the first part of the nephron, and that's where you take the stuff out of the blood. The material that comes out of the blood, we're just going to call it filtrate. It's not urine yet. It's not blood, but it has a lot of stuff in it. Now, how much of this do you get? I said your other capillary beds might lose 7-8 liters a day. The glomerular capillaries lose 180 liters of fluid every day. So if you take all the millions of nephrons in both your kidneys and you measure the amount of fluid pouring into these Bowman's capsules in 24 hours, it's about 180 liters. Huge, huge volume. Again, that's because of the high pressure and the permeability of this filtration membrane. Now, you might think this is a bit of a problem because how many liters of blood do you have? About 5. How could you lose 180 liters a day? I mean, you'd have no blood pressure within, a few minutes. As we'll see, the way the kidneys work as a filter is kind of odd. When we build a filter, we make a screen or some kind of exchange resin. You pour the water or something through it. You pull out the junk you don't want. Only the clean stuff comes out. It's not the way the kidney works. I mean, it works fine, but it's a weird system. What happens with the kidney in this filtration, we're basically taking everything out of the blood. The stuff you don't want, and the stuff you want, and the stuff you need, all the same, it all comes out, ends up in a filtrate. So it's got glucose in there. You don't want glucose washing out with your urine. You might not be able to find a dead antelope to eat next week. You're going to retain that. We've evolved to not lose nutrients in our urine. All of your ions, your sodium, your potassium, that all ends up in there. You don't want that pouring on the ground either. You need all that stuff. The water, you can't lose 180 liters of water. That's mostly what the volume here is water. So the way the kidney works, you dump out everything. Then what's going to happen in the next steps in the nephron, you're going to take all the stuff you want to keep and you move it back into the blood. So I'm going to stop right here at pressure filtration. I'll have to do more with it a little bit later, but that's a long enough video for uploading purposes. So you've got kind of the gross anatomy of the kidney. We know what sort of the nephron is. We haven't looked at its parts yet. We kind of know the blood supply. And again, this afferent efferent arteriole, the reason that setup is there to keep high pressure in the glomerulus. And also you can control it. See, your systemic blood pressure can go up and down a bit. That would have detrimental effects on the kidney's ability to produce urine. If your blood pressure is high, all of a sudden you're producing more urine. If your blood pressure drops, less urine. The blood pressure in here, in the glomerulus, actually stays fairly constant regardless of what your systemic blood pressure is doing. The reason for that, you can play with in quotes, the diameter of the afferent and efferent arteriole. If your blood pressure drops a little bit systemically, you can dilate the afferent arteriole, constrict the efferent arteriole. That will still keep the pressure, the same or high in the glomerulus. On the other hand, if systemic blood pressure gets too high, you'd be filtering too much stuff out of the blood, you can constrict the afferent arteriole a bit. That'll drop the pressure in the glomerulus. So there are mechanisms to maintain what they call glomerular filtration rate. That's the amount of fluid leaking out of the glomerulus into the Bowman's capsule. And that's going to have an impact on, the blood pressure is going to have an impact on the rate of filtration. So by controlling the diameter of those blood vessels, you can control how much you filter to keep it fairly constant. Basically, there's an auto-regulation mechanism. If you increase the blood pressure in the afferent arteriole, that would increase filtration. Some of those smooth muscles, when you stretch them, they'll automatically constrict. So they kind of try and maintain the same flow into the glomerulus and the same pressures. We'll look at that in a little bit more detail later, but I better stop for now, and I'll